Depiction agriculture, general introduction slide. COVID-19 sniffer dog training experimental training studies explore the possibility of training dogs to detect infection using set samples. Training protocols typically involves exposing dogs to samples from COVID-19 positive individuals, sweat, saliva, and urine. Reliable identification, high occurrence rate above 85% have been reported in some studies suggesting that dogs can identify COVID-19 factors like dog breeds, training methods, and samples can influence performance. Copycatting the smell of the depressing captivity scent, research aims to identify the specific volage organ compounds VOCs volts that made up the scent of death. Roles of detective dog. Detective dogs are trained to recognize these specific volts, allowing them to locate human remains even in challenging conditions. Research gap slide. The effectiveness of dogs, how dogs are currently used to detect spotted lanternfly masses, and why this is important. The research gap elaborate on the lack of knowledge about the long term of canine detector and the cost effectiveness compared to other methods. The importance of your research question, emphasize why it's crucial to understand how weather affects dog performance and how trained can improve their efficiency. The potential impact, explain my research can provide evidence-based guidelines for using canine team and controlling spotted lanternflies, which would benefit vineyards and the economy. Research question, how do where temperature ranges 50 to 65 Fahrenheit, 66 to 80 Fahrenheit, and humidity level 20%, 60%, 90%, effective currency percent of correctly identify egg masses, and search time of dogs trained to detect Lycorma, delicatula, egg masses compared to the performance and control ideal conditions 75 Fahrenheit, 50% humidity. Hypothesis trained dogs trained using a positive reinforcement method rewarding a correct detection with treat and praise will demonstrate a significant higher current rate, at least 15% increase in correctly identified egg masses in detecting delicate egg masses compared to dog training using a traditional connection-based method using variable reed planes or leash corrections for incorrect response. Method, the spotted lanternfly Lycorma delicatula is the invasive insect that poses a significant threat to agricultural crops and hardwood trees in North America, Smith 2020. Scent detective dogs have been successfully used to detect their own invasion species. Jones 2018, Williams 2019 suggests their potential of spotted lanternfly detection are finding aligns with previous research indicate the target odor training is an effective method for training dogs method. David 2017. Then I have my graph. The blue one is for detecting and the probabilities for the red one. Then my project timeline. January preparation and baseline, 1 to 2 weeks, finalize experiment design, weather materials, and rescue dogs. Week 34, establish baseline performance for all dogs and divide them in training groups. February, initial training, weeks 5 to 8, begin training session for both positive reinforcement and correction. Base group focus on introducing base detection tasks and initial responses. March, advanced training, weeks 9 to 12, continue training, increasing the complexity of detection tasks, model progress, and adjust training methods as needed. April, assessment, weeks 13 to 16, assess detection occurrency and variation, collect detailed data on correct and incorrect detection of each dog. May data analyze and reporting weeks 17 to 20, analyze data to compare groups occurring rate, compared to a comprehensive report with finding and conclusion. Then my reference page.